know that the phenotype is the physical characteristic, where the genotype is the actual genes that the individual possesses. As the dominant trait is the one that shows up, you have two options for the genotype, right? You can be homozygous dominant or you can be heterozygous dominant. So in order to tell what the offspring of the F1 generation would be, we would need to know how to do a test cross. So, such an individual can either be homozygous dominant or heterozygous. Homozygous, again, meaning that they have the same genes, and heterozygous that they have different genes. So the answer is to carry out a test cross, breeding the mystery individual with a homozygous recessive individual. Now think about why you would do that. The only way that a phenotype for a recessive individual would show up is if they are homozygous recessive for that genotype, right? So if any offspring display the recessive phenotype, then the mystery parent must be heterozygous. So let me show you what we mean by that. So we have our test cross, right? That's what this is going to be. Now, in order to find out the mystery parent, we would need to cross the individual with a heterozygous. Well, we're going to cross them with a homozygous. Let's see. What do we say here? A homozygous recessive individual. So let's just say little p, and the mystery individual, okay, homozygous dominant or heterozygous. So we're going to do both. First, we're going to do homozygous dominant, and our answers here are going to be big P, little p, big P, little p, big P, little p, big P, little p. So we know then that they're heterozygous. All the offspring in the F1 generation are heterozygous, right, for their genotype. But the phenotype, they're gonna show the dominant trait. Even though they possess the recessive gene, the dominant trait's going to be the only one that shows up because the dominant trait masks the recessive gene that is in there. Okay, so now let's switch and let's do our test cross with our homozygous recessive with a heterozygous parent and see what we get. Big P, little p, little p, little p, big P, little p, and little p, little p. Now we know we have a 50% chance of showing the dominant phenotype, and we have a heterozygous genotype, right? And we have a 50% chance of the recessive genotype showing up, and us being homozygous recessive. Okay? So those are Mendel's test squares. And this is what Mendel did. Remember, he looked at the coloring of flowers and pea plants, and we know that the dominant phenotype, we know that's what shows up because the purple flower is dominant, but we don't know the genotype, right? You can either be homozygous dominant or you can be heterozygous, right? And so you cross it with the recessive individual, little p, little p. So if the purple flowered parent is homozygous dominant, this is what our test square would look like, just like we did. We have all offspring are purple, which means they show the dominant phenotype, but they're all heterozygous in their genotype, right? They possess the dominant and the recessive trait, but the dominant one is the only one that is shown. Now, if the parent is heterozygous, then once again, just like we did, you have a 50% chance of the offspring being purple, showing the dominant phenotype and their heterozygous genotype, and then 50% chance that the offspring is white, which was the recessive trait. So you have 50% chance of being homozygous recessive in genotype, and if you are homozygous recessive in genotype here, then the recessive trait is going to show up. That's the only way the recessive trait is going to show up. Now, Mendel derived the law of segregation by following a single character. The F1 offspring produced in this cross were monohybrids, mono meaning one, and individuals that are heterozygous for one character. A cross between such heterozygotes is called a monohybrid cross. Mendel identified his second law of inheritance by following two characters at the same time. He crossbred two true breeding individuals differing in characters to produce a dihybrid, di meaning two here. You're looking at two characters. In the F1 generation, heterozygous for both characters. So a dihybrid cross is a cross between F1 dihybrids, can determine whether two characters are transmitted to offspring as a package or independently, hence the law of inheritance. He was trying to determine if these two characters are passed along to the offspring together or if they separate and then are passed down. So what Mendel did was he looked at two characteristics, right? Yellow and round. This is homozygous dominant. The two Y's being yellow and the two capital R's being round. And then he crossed that individual with a homozygous recessive, which would be lowercase yellow, which represents green, and then the lowercase r and the lowercase r represents it's kind of bumpy is what Mendel called it. So you've got a homozygous dominant individual here with a homozygous recessive individual here, and we get a 50% chance of each in the F1 generation. So you then cross these guys in this dihybrid cross, and you have, remember we said the independent assortment, that each gene goes to one of the sex cells, right, our gametes. So if the sperm is homozygous dominant here versus homozygous recessive here, and your egg is homozygous dominant here versus homozygous recessive, this is your choices. So you have 3 out of 4 chances of being yellow and round with a 1/4 chance of being green and irregularly shaped. Once again, there's his 3-to-1 ratio that he found at the beginning of his studies with the pea plants. So you can go on to look at even more. If you do more crosses, you're going to get a much larger rate. I mean, you're going to get a much larger number, right? You have a bigger population sample. So despite all of these large numbers, you still get this 9:3:1 ratio. So, using a dihybrid cross, Mendel developed the law of independent assortment, meaning that the pair of alleles